All right. Well, you'll see all my co-authors here that I first off want to thank before I forget. This too was work of several people. Go ahead. Oh, wait, am I doing it? So what you see here is the United States and really the Mississippi River watershed. And every year a large hypoxic zone forms right here in the northern Gulf of Mexico, mainly due to these nutrients that come into the water in this huge watershed and out the Mississippi River into the northern Gulf of Mexico right here. So we're well aware that this is happening. So the formation of this zone has been well documented, but the effect on fish and fisheries less so. Also, we have looked into this already, the effect of hypoxia on these fish species of interest and when it's not there. But really in a bit of a crude way, what if there's no nutrient outflow and no formation of hypoxia and compared with it being there? So really the intent of this study is to do it in a more nuanced way, really more plausible scenarios of nutrient reduction and how that affects the hypoxic zone and then the fish and fisheries. So the fish species that you see here is just a small selection of important ones, either commercially. The Penaeid shrimp, the Gulf menhaden are the biggest fisheries by weight and by dollars. A very important recreational fishery is red snapper in this area. Atlantic croaker is the most abundant fish really there, so it represents the ecological important species. So as you saw from the title, this is a co-produced model. Was a project in which we included federal and state agencies. So you see a long list here. I don't think you know any of them really. Some of you do from the United States. So this is just to indicate there were a lot of relevant state and federal agencies that had representatives on an advisory board that really incorporated their thoughts into this project through workshops and a survey that we had during this project. So we've had 3 workshops to kind of get one at the start, in the middle, and towards the end. And we've also this was the work of a PhD student of mine, send out a survey. So I don't expect you to read it. It's just so that you know that there was a survey out to the same advisory panel and with questions that kind of helped us make our work relevant. So I'll start with information learned. You see us, thinking and working on, on what we're getting out of these workshops. So from the start, it was what species to include in the model. It's quite a large model. So we have 60 groups in there, and there was input in there, like what would you really like to see in this model? Also, what scenarios to run with this model? What would be relevant for these agencies? What type of output metric and data visualization is useful? Also, what we learned from them is that uncertainty analysis is necessary. They're not going to accept anything we provide really without it. Part of the workshops, especially in the end, was training. Like, here we have all these models, come learn how to do this. And we've learned from that they wanted a decision support tool and not know how to do this modeling. So a PhD student actually who's here, Michelle Schaefer from Christy Lewis, really focused on that decision support tool. And then what species to include in the decision support tool. We quickly learned that it's soon much too large. You have so much output. So which species exactly, which years, months, it was a very small selection from what was available. So that helped again to see what was useful to them. All right, so as far as the scenario, so this is focused on reducing the hypoxia. A lot of the representatives on this panel were also representative on the Hypoxia Task Force, which is a big group made up of state and federal agencies. And they have goals of reducing the hypoxic area to 5,000 kilometers square. So first had to be determined, what does that mean in nutrient reduction? And then that was by 2035. I can tell you that years shift, so that's not necessarily the year anymore. And then the interim target to reduce it by 20%. So it's kind of two different things. One is a percentage of production. The other one is kind of a size measure of the hypoxic zone. So we went with these as scenarios. What are the effects on living resources of reductions in nutrient loading by 20% and by whatever amount is needed to reach that goal of reducing the hypoxic zone to 5,000 kilometers square. And you can see from here that that's not necessarily what's currently happening in this area. So stuff needs to happen. And this is a coupled modeling approach. And I guess here, the lower trophic model is really the term used. So it's sort of a hydrodynamic model with a biogeochemical module in it. Showed that this can be achieved by reducing nitrogen and phosphorus between 40 and 50%. So we used both those scenarios. So the 20%, the 40%, and the 50% to do our analyses. All right, so model development on my end was creating the Ecopath model. We had 66 groups that included multi-stanza groups, some of it from surveys. We have a lot of surveys in the area, a lot of information from that. Some of it from stock assessment. So any species that had stock assessment, we used the same age classes in the multi-stanza, and we used information from that. We used Ecosim to calibrate the model, and then Ecospace was really the framework of the model. We ran it in the IBM mode, so the individual-based model mode. And this, to get at that uncertainty, this was kind of a new thing at the end, is to do spatial Monte Carlo runs. The Ecopath model. And so there's fisheries in it as well, and all the species and their lines, and you know the drill. Then, of course, we had environmental responses in there. We had, so the hypoxia, like forcing function, or really and spatially, the maps came from the RAMS model. The response curves I got from survey data, because the trawl data of catch, they would also measure, dissolved oxygen temperature and so on. So we created response curves from that. This is the model area. We had to match, of course, the lower trophic model area. So the orange was to make sure we really only had cells in there that could receive information. This is showing really just 1 year of output from the RAMS model. So you can see the Mississippi River here. There's phytoplankton expressed as chlorophyll right here. You can see time here, so it's spring. As soon as summer comes, there's a lot of phytoplankton growing here. When they sink and die, you see the hypoxia forming at the bottom. So the oxygen is on this legend right here. It becomes quite severe in summer. You see it's July now, August, it really forms. And then it really dissipates again when you go later into the year. So this is September, it's still there. October, it's disappearing. So by month, we put this information in the Ecospace model through the spatial temporal framework. So this is just to show some output, like, do our species actually respond to this hypoxic zone? So here we have white shrimp, for example. It's the little blocks because of the IBM. You see the months going, and indeed, when summer starts, they really clear out of that area and then come back later in the year. So it's a spatial issue, but also a temporal issue. It's not there all the time, and we see that in the species response. So when we don't reduce the nutrients, again, to kind of show that spatial response, and also the temporal response, here we've got those 4 species again that I said I would focus on. that in March, you know, you see some traces of that in the spatial distribution, that something's going on there, but it's very different from August, where they're really kind of clear out of that area, and they have different responses to dissolved oxygen, so it's different for each of the species. So we know something's happening with hypoxia, and then we're looking at the nutrient reduction scenarios. So here it's August, because that was when we really saw that strong response, right? So here, if you go on the rows per species, you can see, well, how do they respond then to the nutrient reduction scenarios? And you do see that it's improving. So here too, it kind of depends per species how strong the effect of it is, that they return to that area. But you see there's an effect there. Oh, just real quick, that spatial Monte Carlo, these are probability density plots. It also helped us kind of look at how robust the model was. These look pretty good for the 50% reduction scenarios of what our output that we show is and what kind of the probability of output is in biomass. But interestingly, and I'm using those probability density plots to get these areas of uncertainty around it, and just for a simple picture, what you'd see is just around the baseline what the probability is, and whether those scenarios fall outside of it, because that was really the interest. If we change it, does it significantly fall outside of not changing it? And you see, if we just look at biomass and not a distribution, for the most part, it doesn't, right? So it doesn't change very much. The different colors are the different nutrient reduction scenarios. Black is the baseline. At first, it's just that first run without nutrient reductions, and then we start the nutrient reductions. And then the standard deviation just around the baseline. So to look at whether the model was just not responsive, I also separated, you know, nutrient reduction and hypoxia. If we just reduce hypoxia and keep the nutrients the same, you do see improvement, increases in biomass. So it's really that the nutrient reduction and the hypoxia reduction are kind of counteracting each other, so that the biomass doesn't really change very much. But both types of results are interesting to the practitioners. So they may know now, okay, the total biomass might not change very much, but they're interested in the spatial distribution as well. And this is just a quick shot of that decision support tool, so they can look at it, you know, which species kind of clear out, how does it change with the nutrient reduction scenario, what does it do to total biomass, and they can toggle different situations, and they've been trained in how to use this thing. But as a conclusion, really the distribution is more strongly affected by hypoxia than total biomass of most of the fishery species, and really most species in this model. So those positive effects of the hypoxia reductions are counteracted by the negative effects of reduced nutrients. But the spatial shifts are still meaningful to practitioners, so they are interested in these results, and the decision support tool really facilitates the use of that. I know perhaps the question is going to be the uptake of this information, right? I think right now really a bottleneck is getting those nutrient reductions in place. So the Hypoxia task force unfortunately doesn't have a lot of teeth, so they can't really make it happen. So the year is shifting. So we know what may happen when it does happen, but making it happen right now is really what's the issue. So I'd like to thank my collaborators. You can check out the tool, a first paper on this. There's another one coming, my funders, and I'll take any questions.
Thank you, lovely talk. I was wondering if there is explicit information from the field with respect to whether fish can identify the danger of dead zones and escape, or alternatively stay and die? Do we know something about it?
Yeah, we do. So there's surveys in the area. Also surveys, I guess you don't call it surveys, but information of where shrimp boats, for example, do their fishing. And you can see that they hang out on the edges of the hypoxic zone. So papers that have used data from the field see that there is, a clearing out of that area. Now, it's a combination. So I've used the response curves on the foraging. So we know they can, especially, the bottom feeders, they can't quite reach their food source. So the exact amount of mortality, we don't really know because it's a combination of movement and biomass reduction, which is what we reflected in the model. So we can replicate the patterns, but really like what amount of that is movement and what amount of that is mortality, we don't exactly know. We just know the distribution patterns are replicating.
But roughly most of them escape and less die, or?
Yeah. So, and that's also why I reflected it with the foraging. So, if you're, so I'm doing a lot of work like this now in shallower areas with oyster reefs. There they have no choice, right? So they, when they're stationary, they can't move. So definitely for the nekton species, movement is a large part of what happens, and also why you see a recovery in the months when there's no hypoxia and a small effect on total biomass.
All right, thank you.
Hi, can you hear me? So one question is like from this region, a lot of like the nutrient loading coming from farming activities. Is there like any involvement from like the land-based stakeholders into the decision to process, or are you guys thinking about it, or is it already undergone?
Yeah, so the Hypoxia task force that I mentioned is this group of representative of states and federal agencies, or one federal agency, lots of state agencies. They're all in the watershed. So there's a lot of representation higher up as well on that task force. So they're all involved. There are ideas of best management practices of what to do to get less nutrients in the river. And there's definite work on that. There's not a whole lot of enforcement that they can do to make it happen. So a lot of it is, this is what we should do. So I would say the ideas are there. Really, what would be a nice follow-up study, and I've talked with colleagues about this, is to look at, well, instead of just deciding on a goal, let's, do a 40% reduction. What reduction do you get if we do implement all the best management practices to the best of our abilities? What is that reduction? And then look at those effects, because so far we know, the work is there, the knowledge is there if we need to do all this. What's happening now is not resulting in the reduction scenarios that are set as the goals, for sure.